Hello, everybody. Welcome to Ms. McGuire online video lectures. Today we're covering DNA structure. This chapter covers both structure, DNA structure, and replication. So I will divide this video lecture into two parts. And the first part, we will focus on discovery of DNA and structure of DNA. In the second part, we will talk about DNA replication. So let's go ahead and begin. We're going to begin with learning outcomes for students. So after studying this chapter, you will be able to describe the experiments that demonstrate that DNA is a genetic material and that protein is not. Explain how Watson and Crick deduced the three-dimensional structure of DNA. List the components of a DNA nucleotide. Explain how two chains of nucleotides form a DNA molecule, describe how a long DNA molecule folds and loops, and explain the semi-conservative mechanism of DNA replication. In addition, you will be able to list the steps of DNA replication, explain how polymerase chain reaction is used to amplify DNA outside cells, explain the basic strategy used to determine the base sequence of a DNA molecule, and explain how next generation sequencing improves upon Sanger sequencing. Oh, that's a lot, right? Okay, so let's go ahead. So genetic material must carry out two jobs: duplicate itself and control the development of the rest of the cell in a specific way. So when we're talking about genetic material, what is that molecule that satisfies these two criteria? The molecule that can duplicate, make a copy of itself, and the molecule that will control all other functions inside a cell. And the only molecule that fulfills these requirements is DNA. So let's go ahead and see how our research started. History of DNA. So Frederick Miescher, 1871 or 71, was a Swiss physician and biochemist. He isolated nuclei from white blood cells in pus and found an acid substance that was made with that contained nitrogen and phosphorus. He called it nuclein, and later it was called nucleic acid. Excuse me. In 1902, Archibald Carrad, English physician, linked inheritance of inborn errors of metabolism with the lack of particular enzymes. So that's a lot, right? So look, so let's say if we have a baby that is born with some problem, metabolic problem. So for example, they cannot digest, cannot digest lactose, right? You've heard about people who are lactose intolerant, right? So they cannot digest lactose. So why they cannot digest lactose? Well, this guy, Garrod, he linked this problem with indigestion of lactose with the absence of particular enzyme that break down that lactose, right? You don't have enzyme, you cannot digest something, right? That's interesting. Next, let me see, just making sure I don't skip. Frederick Griffith, English microbiologist that worked with Staphylococcus pneumonia bacteria, which exist in two types. Okay, so let's pay attention because this is very important. So he working with the bacteria that cause pneumonia, right? And he discovered that there is two types. Type S starts for smooth, that is enclosed in a polysaccharide capsule. It just morphology, structure is different, right? And type R, and R stands for rough, that has no capsule. And the Griffiths, what he did, he termed the conversion of one bacterial type into another as a transformation. So he discovered this process and he named it transformation. When one bacterial type, right, became another bacterial type, right? When type R became type S. So how exactly did this happen? So let's look at Griffith's experiment. So Griffiths had, as any experiment should, a control group, right? So he had two control groups and two experimental groups. So for his control group, he took bacteria type R and injected it into a mouse, and mouse is healthy. So type R bacteria is non-virulent, so it does not cause pneumonia and it does not kill mouse. So that's our control. So we know R is invirulent. Then another control was type S bacteria injected to mouse, and mouse dies, right? So type S bacteria kills mice, type R does not kill mice, right? That's a conclusion, that's our control. Now look what he did. So he hit type S bacteria. So this is one that is dangerous, right? So he hits it and destroys. He pretty much kills it. So it killed smooth virulent type S bacteria, dead. Injects to a mice, and mouse stays healthy. That really kind of makes sense, right? Because bacteria is a living organism, and one living organism causes disease, pneumonia, kills the mouse. If you kill bacteria, it's not supposed to kill a mouse, and that's what his conclusion was, and this is what his experiment supported, right? But here's the interesting part. Now, he took type R bacteria, and remember, type R, if you inject, mouse supposed to stay healthy. He took this heat-killed S-type bacteria, and look, and remember, if you use type S, but heat-killed bacteria, mouse should stay healthy. So he mixes them, inject to the mouse, and mouse dies. That is like, it's like really puzzle. It's like, I don't know, some detective story going on over here. So you take type R that is not dangerous. You take now heat-killed type S that is also not supposed to be dangerous, but when you mix them together, mouse dies. So what can be possible explanation? Well, one explanation could be, what is this type S came back to life? It's hard to believe because, you know, if you kill someone, you cannot bring this back to life. You know, dead bacteria cannot be live bacteria again, the same as dead people cannot be live people again. So that's very unlikely that type S bacteria all of a sudden came back to life, right? However, when he took the sample, the blood sample, he found live type S bacteria. So they're pretty much like, oh my goodness, I wonder if he ever thought about discovering like something absolutely unbelievable, that dead living organism can return back to life. If he thought about it for a minute, I doubt, however, right? Because we know that if something is dead, it's dead. So, but then how we can explain? Well, another explanation is, what if you kill the bacteria, right? So bacteria is a cell, it's a bacteria cells, and there is some chemical inside bacteria that you did not destroy. And that chemical was taken in by type R bacteria. So type R bacteria took in that chemical, that molecule that was in a bacteria type S, and that chemical transformed type R into type S, right? What can it be? So if we can figure out what kind of chemical is that, what kind of molecular is this, that can, what it does, that chemical inside the type S bacteria has information how to kill mice. And when it's picked up by our bacteria, it kind of teaches our bacteria, right? It has this information how to kill the mice. That's what transformation is. So today we know it's DNA. I'm going to tell you right now the answer so they know how it was, so you understand better. When you kill type S bacteria, right, you did not destroy DNA of type S bacteria. Now, type R bacteria pick up that DNA, and DNA has instruction, right, how to kill the mice, how to cause pneumonia. But right, so we know that if we go back to very first slide that we started with, I'll go back right here, and says that it should be some molecule that control the development of the rest of the cell in a specific way, control the development and function as well, right? So that's DNA controls the function of the R strand bacteria. That's really kind of like so amazing. But at this time, they didn't know it was DNA. So Griffith had no clue it was DNA. He just knew, well, there is some molecule that is capable of changing type R bacteria into type S bacteria. So it capable of carrying this information, how to kill mice from bacteria S to bacteria R and change this R bacteria, but they didn't know it was DNA yet, right? So next, Avery, McLeod, and McCarthy in 1944, American physicians, they elaborated on a Griffith's experiment. They perform the similar experiment with some change. So they treated this S bacteria that was killed, like heat killed, it's called lysed S bacteria, with two enzymes, protease and DNase. Now, protease destroyed proteins and DNase destroyed DNA, right? So they, okay, so we will get back over here. Let's look at the experiment first, what they did. Okay, so remember how Griffith, he killed type S bacteria. So here we have this heat-killed type S smooth virulent bacteria. So bacteria cell is open. Now we don't have living bacteria, but we have either, we have both DNA molecule and proteins, right? So let me just draw. So we have DNA, we don't have bacteria, but we have DNA molecule and we have some proteins. Let's put them in a like circular structure, right? Now, trying to see my red color. Now he used protease, right? If you use protease, what protease does? Protease destroyed proteins. So they kind of like, okay, so let's just kind of destroy all these proteins, right? We know we don't have proteins. Now we have only DNA. Now what happened if we use protease? Then the R type became S type and mouse dies, right? Because we have DNA. Now look what in next example what happened. In the next example, here again has DNA, right, and proteins. But this time, they use DNA, so they destroy DNA. So now they have only proteins. So they mixed these proteins with type R bacteria and mouse leaves. So type R bacteria, if proteins, it take in proteins, but proteins do not carry this genetic information, how to be virulent, right? That makes sense? So let's just make sure that we understand it completely over here. So if you have mixture of DNA and protein, and you're wondering, what is that molecule that transfer information from S to R? What is that molecule that is capable of making type R virulent and kill the mice, right? So if you have both DNA and protein, what you need to do? First, you destroy all DNA and you have only proteins, and you see, nope, if I don't have DNA, nothing happened. But if I destroy proteins and I keep DNA functioning, right? So I keep the structure and shape of DNA, then DNA transform R into S. So that's what the Avery, MacLeod, and McCarthy did. So only DNAs prevented transformation. That means if you destroy DNA, no transformation. Such DNA is transforming principle. And DNA can convert type R bacteria into type S bacteria. Okay, well, now we know that we have a molecule, right? Molecule that is capable of regulating a cell activities in the way that let me put this way. So there is a bacteria that there is a molecule of DNA that regulates all the activities of cell, right? It makes this bacteria either virulent or harmful, or non-virulent and benign, right? So that's a DNA molecule that has this information how bacteria should function, right? But we needed to support this statement that DNA is a molecule of inheritance, and Alfred Hershey and Martha Chase in 1953 provided more data to support the hypothesis that DNA is hereditary molecule. They were American microbiologists, and they used this time E. coli bacteria infected with a virus that consisted largely of protein head and coat surrounding DNA. So let's look again at the experiment, and then we will get back to this slide. So here you have a virus, and we know virus is made of protein coat, so that's a protein coat, and I don't want it to be red, so let me go back to blue. So coat is made of protein and it has DNA inside, so we pretty much have both DNA and the protein. And what they did, they marked in one experiment protein with radioactive element, and in the second experiment they marked DNA with radioactive element. So they marked protein here with radioactive sulfur, and they marked DNA with radioactive phosphorus, right? So here we have radioactive proteins. So they mixed virus with the bacteria, and they know that when virus infects bacteria, it's deposit some part of it inside the bacteria, right? Some part of the virus goes inside the bacteria, and it pretty much destroys the bacteria, or kills the bacteria. Right, so when they marked the protein, the bacteria was not radioactive, that means that virus is not injecting protein inside the bacteria. But when they used radioactive DNA, they could see that, you see how these little viruses attach to the bacteria, right? And bacteria is not radioactive in the first experiment. And here little viruses attaches bacteria. And then what they do, they actually injected their DNA inside the bacteria, and bacteria is radioactive. So they know virus injected its DNA inside the bacteria. And that DNA, viral DNA, has the information that tells bacteria to start making more viruses, right? Because what virus does, virus reproduces inside, and this specific virus is called bacteriophage. This is the virus of bacteria. So virus reproduces inside bacteria. For virus to reproduce, it should be instruction how to make more viruses. So the virus, from one virus, we can go to many, right? So virus reproduces, right? So we have more viruses. So now we think that DNA has information about the structure of virus and function and how to make new viruses, but how we can prove it, right, that it's DNA. And that's why they marked DNA with radioactive phosphorus, and they knew that virus specifically inject DNA inside bacteria, and viral DNA has information for bacteria now how to make more viruses. So that means DNA is the molecule of inheritance. So that's what they did. So they used E. coli bacteria infected with a virus that consisted largely of protein head and coat surrounding DNA. They grew a bath of virus in a medium containing radioactive sulfur and phosphorus. And blender experiments showed that the virus transfers DNA, not protein, into a bacterial cell, and thus DNA is the genetic material. The experiments that revealed the structure and function of DNA looked back and forth in time. And why? Because they came from different disciplines. If you look over here, they came from microbiology, from physics, from physics again, from biochemistry, right? So that was physicists, biochemists, microbiologists, because the molecular genetic did not exist yet by that time. Okay, so here we have another Russian-American scientist, biochemist, that is, his name is Phoebus Levene, and he identified that, so he was, he was pretty much like a pioneer in discovering of DNA composition, right? What is this DNA made of? All the previous experiments that we described, what those experiments provided us, they provided us with information that DNA is the genetic material. But what is the structure of DNA? So Levene identified that 5-carbon sugar ribose and deoxyribose are part of DNA, right? And it happened in 1909 and 1929. He revealed chemical distribution between RNA and DNA. So RNA has ribose and DNA has sugar deoxyribose. He discovered that the 3 parts of nucleotide are found in equal proportions within DNA. So in the DNA, you have equal proportion of sugar, phosphate, and bases. And deduce that a nucleic acid building block must contain one of each component. So he pretty much, like, if you like, how am I supposed to remember this? Just the main information on this slide is that if you look at chemical composition of DNA, it's made of sugar, phosphate, and base. And sugar, phosphate, and base are in the same proportion, in the same ratio. So you have one sugar, one phosphate, and one base. Or you have two sugar, two phosphate, two bases. Or you have a million sugars, million phosphate, and million bases. So then the Levene proposed that it should be a building block then that has all of these components. So it should be one building block has sugar, phosphate, base, another building block, sugar, phosphate, base, and you put million of these building blocks together, and this way you have the same proportion of sugar, phosphate, and base within DNA molecule, right? Then Erwin Chargaff in 1951, Australian-American biochemist, he analyzed composition of DNA from various species and observed regular relationship. So when he looked, Levene said, well, it should be a building block. And really, this building block is nucleotide, right? So DNA is made of nucleotide. Let me just draw it for you so we can have an idea. So DNA is made of a building block called nucleotide, and each nucleotide is made from sugar, base, and phosphate, right? So that's one nucleotide. And then you put so many of these nucleotides together, and that's how DNA is made, right? So we know that nucleotide is made of sugar, phosphate, and base. And now when they look at those bases, right? When they look at those bases, this is what they noticed. They noticed that if you take DNA from different species, you take it from plants, from animals, from fungi, from different animals, from different plants, when you analyze it, and you will see that those bases, when you take an amount of adenine and guanine, it's always equals the amount of thymine and tyrosine, right? So always amount of A bases always equals amount of T bases, and amount of C bases always equals the amount of G bases, right? So that's what Chargaff discovered. So this is just set the stage for further analysis and finally to discovery of DNA structure, right? So it's all get together. And I will get back to it and we will sum up everything. Then we have Rosalind Franklin and Maurice Wilkins in 1952, English scientists. They use a technique called X-ray diffraction, and they deducted the overall structure of the molecule from the patterns in which the X-ray were detected. So they shoot this X-ray into a molecule of DNA, and they get a reflection from DNA molecule, and they analyze this reflection pattern. And they distinguish two forms of DNA, A form, which is dry and crystalline, and B form, which is wet and cellular. So B form, this is the form of DNA that present inside living cells. It took Franklin 100 hours to obtain 451, very famous photo of the B form of DNA. And this is how it looked. And this is our scientist, Rosalind Franklin. And that's her X-ray photo of DNA molecule. So Franklin reasoned that DNA is a helix with symmetrically organized subunits. Now, what do we know? Look, we know that DNA is made of subunits, and each subunit has sugar, phosphate, and base. So this subunit is called nucleotide. So DNA is made of subunits called nucleotides. Each nucleotide has sugar, phosphate, and base. When we look at the bases, the amount of A bases always equals the amount of T bases, and amount or number of C bases always equals the number of G bases. Now we know also that those subunits, those nucleotides, are organized in a symmetrical helix because of the Franklin photograph, right? And here we got them. James Watson and Francis Crick in 1953, American biologist and English physicist. They did not perform any experiment, but rather they used their earlier research that we just discussed, right? And in inferences from model building with cardboards and cutouts to solve the structure of DNA. So those are, I would say, was at that point, they were theoretical scientists when they just analyzed all the information that was available, right? That we just said that we have subunits and subunits made of sugar, phosphate, and base, and we have 4 bases, and they kind of complementary to each other. So number of A bases and T bases are the same, C and G bases are the same. They also look at the famous picture 51, where DNA is a helix, and they use a cardboard, they use some metal parts, and they build a DNA structure. And on this picture, it's a very famous picture. You can see the model of the DNA. Right, they were rewarded Nobel Prize for their discovery. Right, so here we started with Frederick Miescher that isolated nuclein in a white blood cells, then Frederick Griffith that transferred killing ability between types of bacteria, Avery, MacLeod, and McCarthy that discover the DNA transmits killing ability in bacteria. Hershey and Chase that determined that the part of a virus that infects and replicates is its nucleic acid and not protein. Then we have Levene, Chargaff, Wilkins, Franklin that discovered DNA components, proportions, and positions, and then James Watson and Francis Crick that elucidated DNA's 3-dimensional structure. So they build this 3-dimensional structure, discovered 3-dimensional structure of DNA. And then James Watkins later in 2008 has his genome sequenced. Right, so that's amazing history of DNA, study of DNA as a core of genetics. The structure of the genetic material can be considered at several levels. So so if you're looking at genetic material, you can look at bits of information that will be DNA bases. You can look at genes and chromosomes, or you can look at genome part that is loop, fold, touch, and interacts as genes are expressed. So we can look at DNA at very, very different angles. So now what is a gene? Gene is a section of DNA molecule. It's a sequence of building blocks that specifies the sequence of amino acid in a particular protein. So to make it kind of easier, gene is a part of DNA that codes for protein. So gene has sequences of nucleotides that have an instruction for your cells to put amino acid in a specific sequence to make functional protein. The activity of the protein impacts the phenotype. So that's a proteins that make you who you are, right? And your DNA just has instruction how to make those proteins. Because when we think about genetics, we always think like, oh my goodness, the DNA is the most important molecule, and it is. So DNA determines all my phenotype, how I look, how I walk, how I, what diseases I have. Yes, it's all in your DNA, right? But that's the proteins who make it possible, who make your phenotype to be expressed, that causes the diseases or different conditions. In genetics, they have this saying, DNA has all the glory, proteins do all the work, right? So DNA is, what DNA is, it's instruction. It's like a huge cookbook, right? Your cookbook has instructions how to make different dishes, right? The DNA has instruction how to make every single protein in your body. And then those proteins that regulate the activity of your cells, how your cells functions, what your cells do, and how you look, like determine your phenotype. So let's look at the structure of DNA that was discovered by Watson and Crick. So a single building block of DNA is nucleotide. Each nucleotide is composed of sugar. And if you're talking about DNA, it's a specific sugar called deoxyribose, a phosphate group, and nitrogenous base, one of 4 types, adenine, A, guanine, G, they are purines, cytosine C, and thymine T, pyrimidines. So here you can see the structure of one nucleotide, and that's a building block of DNA molecule. So we have sugar, deoxyribose, phosphate group, and this is nitrogenous base. So this is an example of a nitrogenous base, guanine. Now we have 4 nitrogenous bases. We have guanine and adenine, that is A and G, right? A and G, and they are purines. So they have double ring, and here's where sugar is attached, right? And we have pyrimidines, cytosine and thymine, and they are single ring structure, and here's where we have sugar attached. Right, okay. Then nucleotides are joined into chains. Phosphodiester bonds formed between deoxyribose sugar and the phosphate. And this creates a continuous sugar phosphate backbone. So here we have sugar, remember, and we have either purines or pyrimidines. So that's our bases. That's a base. And they're hanging kind of on the side of DNA molecule. And a sugar also has phosphate group, right? And we have this connection between a phosphate of one nucleotide. So here's my nucleotide, right? So here's a nucleotide. So it has phosphate, sugar, and base. So we have connection between one nucleotide and another nucleotide through this phosphate and sugar phosphodiester bond. And we form sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate backbone of the DNA molecule. 2 polynucleotide chains align, forming a double helix. The opposing orientation, head to toe, is called antiparallelism. The head-to-toe configuration derives from the structure of the sugar phosphate backbone. So don't worry if you don't understand yet. So let's look at this structure of now double helix. Let me just draw instead of having my pens that disappear. So here's one strand of DNA, right? And here's another strand of DNA. DNA is double helix, right? So we need 2 strands. Now look this one. Here's our phosphate, sugar, phosphate, sugar, phosphate, sugar, right, backbone. But look at over here. If you look at the beginning of DNA, in this situation, we start with sugar. So sugar, phosphate, sugar, phosphate. So one strand runs this direction from phosphate to sugar, and another strand runs in the opposite direction. And this is what called anti-parallel, right? Like those zebras, right? So one you're looking to the back and one you're looking to the front. So that's how your DNA is. Right? So it's anti-parallel. So antiparallel nature of the DNA double helix becomes apparent when the carbons in a sugar are numbered. Carbons are numbered from 1 to 5, and we start numbering from the base, right? So remember, that's our base over here, and this is our carbon. We number it 1, and we go counterclockwise. So 1, 2, 3, 4, and 5. And that's a carbon 5 over here. And where carbon 5 will be connected to a phosphate. And carbon 1 is connected to our base, right? So we just number those carbons. That makes sense, right? Now look how we now put stuff together. So if you look over here, the phosphate, remember where we have phosphate, we have carbon number 5. So it's a 5' end. And I remember it because 5 and phosphate kind of, well, it's phosphate starts with PH, but the sound is F, right? So 5 and phosphate. So where we have phosphate, this is our 5 end, right? So we go from 5 to 3. So we go 5, then we go to 3 where the sugar is. 5 to 3. And over here, look, we start with sugar. So here we have from 3 to 5. So one runs direction 5 to 3 here, and another, here's our phosphate. So this is our 5'. 5 to 3 goes in opposite way. Right, so note that one strand of a double helix runs in a 5 to 3 direction, and the other strands runs in a 3 to 5 direction, right? So we start with 3, carbon number 3, and then it runs to 5. Right, so that's how this double helix is anti-parallel. And it will be important when we start talking about DNA replication. The key to the constant width of the double helix is a specific pairing of purines and pyrimidines via hydrogen bonds. The complementary base pairs are adenine and guanine and cytosine and thymine. So mnemonic is apples in the oil. Okay, A pairs with T and C pairs with G, right? So they just said that complementary base pairs are, I guess, adenine and guanine always pairs with cytosine and thymine. So that's how it is. So it's a little bit confusing even for me, like, what is over here? So adenine and guanine always pairs with cytosine and thymine. So hydrogen bonds hold the base pair together. So let's look at the diagram to understand it. Right, so here we have adenine, right? And we have guanine, and they are purines, so double rings. So double rings always pairs with single ring, right? So double ring with single ring. And adenine always pairs with thymine, and cytosine always pairs with guanine. And mnemonic over here is apples in a tree and cars in the garage. And A with T, it forms double bond. It's a hydrogen bond. It's a weak chemical bond between a hydrogen molecule and more electronegative molecule, for example, oxygen or nitrogen over here. So this is hydrogen bonds. And guanine and cytosine, they form triple hydrogen bonds between those bases. So, and you can see here, A always pairs with T and C with G, apples in the tree, cars in the garage. Scaffold protein form framework that guide DNA strands. So look, we have this DNA strands, right? We just said about it. We have this strands of DNAs at 5 to 3 directions, 3 to 5 directions, the antiparallel. But we also have proteins that form framework for DNA folding. The DNA coils around proteins called histones, forming a bed on a string-like structure. I'm sorry, not bed, bead. Bead on a string-like structure, like you have a bead, string, bead, like a necklace, like this. So the bead part is called nucleosome. DNA wraps at several levels until it is compacted into a chromatid, and chromosomes substance is called chromatin. And I will explain it more. So let's look at the next slide first. So DNA is a pretty large molecule, and you need to feed this molecule inside a microscopic cell. So let's look. So that's why DNA is highly condensed. So here's our DNA molecule. It's a double helix, right? That's made of these repeating blocks called nucleotides. Now, the DNA then wraps around these proteins called histones. And so we have like several histones and DNA wraps around them. Now, this structure is called nucleosome. So this is our beads. So you see how this our beads on a string, right? Like a necklace, like we already said. So here's nucleosome. Then we have linker proteins and nucleosome again, linker protein, nucleosome, like this, right? So, and the DNA is, where's the DNA over here? DNA is wrapped around those histones. And now when we have DNA wrapped around histones, we call it chromatin. And when chromatin condenses, all right, then it became chromosome. And so we have DNA. DNA wraps around histones called chromatin. Chromatin condenses, became a chromosome. And when chromosome is duplicated, it's made of two sister chromatids. And we will talk about it more. So right now, let's just kind of, from this diagram, just remember that DNA is wrapped around histones, forming a chromatin. Those beads that made of DNA and histone called nucleosome. When chromatin is loose, not condensed into chromosomes, right? Because this, what is chromatin again? Chromatin is just DNA around histones, and it's in a loose form. And so when it's loose, not condensed into chromosomes, then it forms loops at about 10,000 places in the genome. The anchor protein, called CTCF, brings together parts of the DNA sequence within the same long DNA molecule to form the overall loopamy structure. So chromatin forms loops. So look at this picture. Now, what do we have over here? Is this DNA? Yes and no, because it's DNA that is folded around histone. So that's a chromatin now. So it has both DNA and proteins called histone. So that's a chromatin. And now look how chromatins is form those loops. And those loops pretty much hold together. You need to hold this loopy form. And this form is held by the special protein called CTCF anchor proteins. So that's how your DNA look like when it's inside the nucleus and when it's not condensed into chromosome, right? It has this loopy structure or loopamy structure, right? And and this is how it exists inside nucleus. So you can see over here our loops, and that's what make that's a loose in the form of chromatin, inside the nucleus, and when chromatin condenses, and we have chromosome. Okay, so I think that's it for this lecture, because the rest of this PowerPoint is explain us that explain us how DNA is replicated. So I will record another video because I don't want this one to be extremely long. Thank you for watching, and I will talk to you next time.